little green fluorescent protein around the rim. You can see on that bottom animal. What do you think? Yeah.
All right. Good morning. Welcome to Divestream 862. This is the first dive of our new ongoing cruise, searching for the Asgard. And you'll note that this is a very shallow dive. We are on bottom at just over 62 meters depth. Expected to be a pretty short dive and largely focused on some push core samples. But we just touched down, we fired off some niskins. Now we're going to scope around a little bit, settle down, grab our push cores. And we're currently just looking at some nice zooms. So I'm going to turn up the rest of the mics in here, and you'll hear the scientists.
But I don't know what the original substrate is. It's kind of hard for me to call. We get some good frame grabs?
Yeah, I'll get a couple more.
Thanks. Great lighting. That is brilliant. It's going to be interesting coring here. Yeah. Yeah, it looks very sandy.
Yeah, it's making me
Yeah, it
nervous.
might fall out, huh?
Mm-hmm. Try.
You want to look at some other critters?
Are we ready to? Oh, there's a.
Oh, hey.
4D. You say the word, Brett. We'll get started with the coring.
Just watch this guy snore a little bit.
Are we live?
We are live.
All right. Welcome to the dive stream and to dive number S0862, the inaugural dive of the Symbiotic Partners of Asgard research cruise. We are on Station 1, approximately 125 kilometers off the coast of Uruguay. Here in the control room with pilots Zach and Tyler, logger Ian, and me, Jacob, as the watch lead, plus much of the science team of the expedition. We're at a shallow site on the continental shelf at about 62 meters. And our objective today is going to be coring sediment to recover samples, ideally, of Hodarchaea, a group of microbes that we'll give you a little more detail on later, and we'll explain why we're after them in particular. Nice sea cucumber.
Is that what you were saying earlier, looked like gnats?
That is what we were looking at, and they look to me now like detrital flocks. I mean,
Yeah.
yeah.
This stuff's settling.
Junk, sea junk?
So we're just doing a site survey now, and starting in a few minutes, we'll attempt to take our 18 sediment cores for this dive. That's my best guess. Looks like some kind of a sponge. Yeah. Yeah, so here we see the bottom covered by anemones, a type of cnidarian or jelly. These are not technically true anemones. They're probably more closely related to corals like you would find on a reef. My guess is that they're eating this abundance of particulate matter. A lot of which is probably a result of our being so close to the mouth of the Rio de la Plata. We've got a lot of people on the chat.
Well, that's even more.
Bienvenidos. Gracias para juntando la inmersión inaugural de la campaña Spark. Estamos unos 120 kilómetros hasta la costa de Uruguay, en la plataforma continental, en aguas que tienen mucha influencia del Río de la Plata. Sí, hay científicos uruguayos a bordo. Hay Hay unos aquí en este cuarto. Cecilia Alonso de Ulalar.
Hola, buen día para todos. Bienvenidos.
Definitely cucumber.
Anything else you want to see?
Do we want to do any other survey before we start collecting?
Give it a shot. Start coring. Let's see.
Give her a whirl.
Yeah.
Yeah.
Just get the bubbles out of that camera. Alright. Coming out with starboard ram first. You got a preference, starboard, starboard? Doesn't matter. We're doing them all, right? So,
I think
think what?
that's fine. Starboard first.
Don't matter to us.
Do the backups first.
We're doing them all, right?
We are doing them all.
That one.
Cecilia, ¿cómo se dice core? ¿Cómo se dice sedimento?
Do a hoop. Tester first.
Testigo.
Normalmente le decimos core también, pero la palabra correcta es testigo de
You
sedimento.
want the lowers on? Give you a little more light, or you think that just smoke you out, man? Yeah, I got 20% down today. Too much.
You got a shout out on that.
Wow.
Hola, Cecilia.
There's 24.
Sí.
And the
Vamos a explorar sobre la costa uruguaya. Hace casi 200 kilómetros hasta la costa. Gracias para tu paciencia con mi español. Es por eso y otras razones porque tenemos científicos uruguayos aquí en el cuadro de control.
No, en esta campaña no tenemos gente de Argentina.
Yeah, that's great.
Es
Just a
un
bunch of
equipo,
sand.
somos un equipo de 26 científicos
Oh,
a
that's
bordo.
beautiful.
Habemos 4 de Uruguay y el resto es de distintas partes del mundo. Jacob es de Estados Unidos.
Alright, so he's hitting a bottom there of some kind, or I don't know if it's rock or what. But,
Okay.
Okay.
You happy for him to try to come back out?
Yeah.
Okay. Go for it, Tyler. Easy.
That's good.
Okay, so it is coreable.
That's good, man.
Large sample PC9.
Alright, you want us to just load in the next 7 and pull them out one at a time? Guys happy?
Why don't you load in 7, yeah, and then pull in, yeah. Yeah.
Can we approve it for this one?
Yeah, go for it.
Acá Nidia está preguntando si venimos a descubrir los orígenes de la vida compleja como la conocemos en nuestro planeta, que se generó a partir de la asociación de dos células muy pequeñitas que pertenecen a dominios muy diferentes de la vida. Una del dominio bacteria, que de pronto es lo que estamos más acostumbrados a conocer, y otra del dominio Archaea. Y justamente la bacteria hace bastante tiempo que creemos que sabemos cuál fue, pero la Archaea hasta hace poco tiempo era un enigma. Y bueno, estamos acá buscando a esas arqueas. A ver si las encontramos en nuestro mar, que tiene muchas características que nos hacen pensar que sí.
So that push core looks a little different. It just has a strip of tape down the side to seal some holes that are going to be used for sampling from within the core. So we have to have that tape there to keep the vacuum, but afterwards it'll get removed and then you have those little sampling holes.
That's fine. But it's very similar.
No idea. That one went in quite deep.
Yeah.
So just like do them all like that, Tyler. All you gotta do.
Is that one not the core with the holes
It's not, but we threw you a curveball.
That's all right.
It's
So what's
a-
this one that I didn't see?
Yeah.
It's the one with the tape on it.
So under the electrical tape, which
Yeah,
you're
yeah.
right, is to protect something else.
Okay.
There are these special tapes that are installed on the inside of the tube,
Ah, okay.
and they give off fluorescence. Basically, their optical properties depend on the oxygen and the pH inside the tube, and because they're strip-shaped, we can bring it back aboard, put it in front of a special camera,
Yeah.
and ideally, you get the entire vertical profile of oxygen and pH in a single shot.
Oh, interesting.
Very
What kind
cool.
of camera?
It's like effectively a modified CCTV camera, but it has filters for a couple of pretty long wave emission wavelengths, and we
Yeah.
shine excitation lamps. Like,
Reddish.
yeah. We shine UV on it, and it picks up in the infrared
Yeah,
spectrum.
we'll try to get some like photos of that so we can put those up sometime for the stream.
We're hoping to get some UV lights on Sebastian's.
Yeah.
Oh, that'll be fun.
Do we have the push cores that have the little holes on that I saw
Yes.
getting prepped? Okay.
Yes.
I was like, I swear.
And we have two that we know of.
All right.
That's the only reasonable guess that you could have for what was happening.
And you think they always have holes in them, right? Just don't like push car with no holes, not big deal. Kind of zoomed in that little too. So, we got new bulkheads in for 'em, right?
Mm-hmm.
Estamos en un lugar en la plataforma continental sobre Uruguay, unos 120 kilómetros hacia la costa. Por la mayoría somos una campaña biológica, pero por qué los microbios que estudiamos viven en asociación con el sedimento. También tenemos Galen, un geólogo en nuestro equipo.
I didn't get much of there at all.
No, that was pretty
Hmm.
shallow. We already moving the sub around.
Oh,
Did that not have the
no hose clamp.
That's why we packed a couple extras.
Oopsie.
I should watch out a little for them.
Which number was that?
15.
Few folks noting that we are quite shallow. Yes, indeed. In fact, those of you familiar with the floats that we put on our umbilical, the floats are still on the surface. So, quite unusual for us.
Nice. Okay, pushing in. Oh, it's so nice not having all those on the row there, just dragging.
What are we doing? So, specifically, at the moment, we're coring sediment in search of a group of microbes called Hodarchaeales. In general, we're after a group of microbes known as the Asgard Archaea, which are thought to be our closest microbial relatives. So, a couple billion years ago, when very simple cells like bacteria that don't have compartments gave rise to complex cells like our own, which have a nucleus and a mitochondria and all of those other fun parts. It happened because one type of microorganism, an Asgard Archaean, formed a symbiotic relationship with another type of microbe, alpha proteobacterium. And we're in search of some of the living microorganisms that are most closely related to those original symbiotic partners that led to animals, plants, fungi, and us. So pretty much any kind of life that you can see, for the most part, is going to be made of what we call eukaryotic or compartmented cells. It's trees, fish, humans, flowers, mushrooms, kelp, you name it.
All right, you guys have a first to pick these up?
Yeah, I think so.
Pick 'em up.
Are you happy with the state of all those cores? Great. Let's recover, please.
Sure enough. Let's see what's how far out there. Let's do that. Interesting.
interesting to see how much DNA we get on these. I mean, I don't think it's gonna be a problem.
It'll be interesting to see how much of that DNA is from sunken algae.
Yep. Yep.
Ian, please mark PC16.
Just your ones. Look like you're gonna say something.
Alright, all set?
Thank you.
What say you? Yeah. Well, there's definitely plenty of biomats, so. Yeah. I mean, such illicit. Asgard, yeah. First what?
Billones
Yeah.
de años.
Yeah, so that's very similar. The what? Highest.
Estamos más que 100 kilómetros hacia la costa de Uruguay en la plataforma continental. Can we snip those or do they have to be pulled off, the zip ties?
The zip ties, so that's so when they go in, they hold the little valve open so that the bubbles can escape.
Right. Is it? Yeah. No, go
You'll see on
ahead.
the other side, when he pulls them out, they actually come out of that washer.
Oh, okay.
Is that
Got
what
it.
you're asking what they're for?
Got it. Well, I was asking what they're for and whether they can be snipped, but it seems like they're sticking out intentionally.
Yeah.
Yeah.
Otherwise, we'd have a lot of air stuff in here.
Alright, I got PC14.
Got it. Thank you.
So that big old push cord. I want to see if it a different color at the bottom.
Yeah.
Maybe get some clay or something.
Maybe.
What do you think the bioturbation's like here, Ian?
I don't know, there's a bunch of stuff digging into it, so
Yeah.
probably fairly decent.
Vocab word of the day, bioturbation, is mechanical agitation of sediment by animals that live in it. Nice. Okay, let's mark PC13, please.
Alright, PC13 logged. Jacob, I think I was closest to my temperature guess on the bottom, but I busted because I said 12 and you said 9, I think, right?
Hmm.
What is it?
11? Yeah, that's fair.
Something interesting in that one.
Yeah. It's white.
Calcareous.
Nice corn deep.
Mark PC12, please.
PC12 Bart.
You were on that last one when we did like 16 at the beginning of every dive, right?
Yeah.
Got some practice there.
I was gonna say, this was just really familiar.
You can see the little white sensor strips peeking out the bottom.
Oh, yeah.
Yep. Because they're light reactive, they can actually be damaged by really bright light, hence the tape.
That's why the tape.
Yeah, because the vehicle lights are bright.
Or sunlight, I guess.
Oh, yeah.
Sunlight, you probably heard him too, huh?
Oh, certainly. I mean, probably worse than the vehicle lights, but generally that's not a concern. I mean, I suppose when we're on the surface. The strips are designed for experiments with things like plants and you know, petri dishes full of bacteria. As far as I can tell, nobody's actually used them on a sediment core before.
Really?
So this is kind of a trial run.
But I guess they don't have any problem with seawater then, huh?
No. Yeah.
That's very cool.
Yeah, as long as if you calibrate them right, they can work in air, they can work in water. I just couldn't. We searched far and wide and couldn't find anyone who had stuck them on the inside of a core sleeve.
Whoa, you guys are doing groundbreaking stuff.
Stay in there.
Boy, that's a nail biter.
Oh, she's gonna go. No.
Do you want to call it or and repeat or go for it?
No.
Okay.
Put it in there. Get it in there. Quick.
No pressure, Tyler, but go, 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 go.
Oh, there it goes.
It's boiling out.
That's all right. Actually, pull it down, maybe. Yeah. See
Yeah.
if you can.
I was afraid we'd end up dumping it in the
Yeah.
quiver.
I think we dumped a little bit.
That's all
Yeah.
right. Shake it.
We'd go for that one, or?
Hey, you know, 1 out of 16 isn't a bad.
Pretty
job.
good.
No, yeah, it's great. Oh,
Just
it's
that.
cleaned out.
I saw that sand, I was like,
Yeah, totally. I didn't think we were gonna get this many cores.
Should we hit it again?
Yeah, let's try it again.
This
Yeah.
is number 10.
Put it down a little bit closer. Let's do it.
Yeah. Oh, you're right. I don't think I called out PC11.
Yeah, I
Thank
got you. Sorry, I'll
Appreciate it, Jimmy.
Oh, that's even better. Look at that.
it's your destiny.
Yep, exactly. Blessing in disguise. Oh man, could be the best core.
Hmm?
Tyler's mad at it now. Well, it has the white stuff in there too.
Cool. Great.
Beautiful.
Let's call it mayonnaise. It's probably mayonnaise.
Aioli.
So yeah, it's probably got a little bit across the bottom. but
Yeah.
Mm-hmm.
Oh, that's pretty good. Large sample PC10, please.
All right. PC10.
At 1000 UTC on the button.
Push down the other tube here, Zach.
What's that?
Alright, let's see if I'm gonna shut it down on the ball.
Oh, sure.
Just to make sure it doesn't come out of there or something.
It's full, right?
That's a lot.
There we go.
Time to start the day.
So to reorient anyone who has just joined the stream, we're coring seafloor sediment off the coast of Uruguay in search of our nearest microbial relatives, the Asgard Archaea, many of which live in close association with sediment, which is why we are collecting so many cores, these tubes full of sediment.
Blake.
All right.
We'll just do those two last, huh? The two with the holes in them.
Is that okay with you, Jimin? If we do the, is it all right with you if we do the pour water cores last?
Yeah, yeah.
Okay.
It would be great if we could be near a non-pour water core.
Great.
Be near a what?
Be near a non-pour water core. Would it actually be possible to lay out the last 10 cores before recovering? And that way we can
Where you can see them all next to each other?
Exactly.
Yeah, sure.
Thank you. Actually, the entire habitat off of Uruguay is relatively unexplored, as I understand. There was one other Falkor cruise in this area recently. But we'd expect to be able to find these microbes in a lot of coastal, nutrient-rich areas of the world's oceans. So I think that exploring this area is sort of a bonus, in that anything we find is going to be novel, because these areas haven't really been microbiologically explored. But we also have a pretty reasonable expectation of finding the specific bugs we're after.
Have y'all done cores like this with an ROV before?
I, Jacob, have never done cores before.
Really?
Yeah, I've done midwater work, I've done hydrothermal vent work, but this is a new adventure for me. Obviously, this is not the case for Brett, but I think, and we may hear from Brett shortly, all of his work has been with human-occupied subs, like Alvin.
Okay.
Correct. The geologist in the room is Galen Halverson.
Estamos en la campaña buscando a los Asgard, que arrancamos ayer desde el puerto de Montevideo y vamos a estar hasta el 3 de diciembre en el mar uruguayo.
That starfish scuttling along here.
Yep.
That would be a eukaryote. Why do we even have phyla in the annotation? We should just have domain.
You're right. Yeah.
Did we mark a biology observation?
Oh yeah.
Thank you. So just for tracking purposes, we're nearly a half hour into this operation. And probably somewhere near halfway through.
Yeah. Going smooth.
Para quienes preguntan dónde estudiar en Argentina para hacer biología marina con especialización en tiburones, no estoy muy segura, pero creo, por lo que entiendo, que se puede estudiar en la Universidad de Buenos Aires y en la Universidad Nacional del Sur, ahí en Bahía Blanca. Y no sé si hay especialidad en tiburones, pero haciendo una licenciatura en oceanografía es posible después especializarse en distintos temas. De hecho, en esta campaña, aunque no es el foco central, como ven, el foco central son los microorganismos, de todas maneras, desde el equipo uruguayo estamos complementando con diferentes líneas de trabajo, y uno de los investigadores, que se llama Rodolfo Vogler, justamente es especialista en condrictios, que se llaman, que son los tiburones, las rayas, los torpedos, y él va a estar analizando las imágenes que captemos con el Sebastián, justamente para buscar a este grupo de organismos. Así que bueno, hay, si bien no está a bordo, hay la posibilidad de intercambiar luego al respecto de esta parte de la fauna.
Sometimes it does that and it goes away at depths, but obviously we wouldn't know that here. We might know that in the next spot.
What do you think will go away with depth?
These ground faults we have on surface sometimes, like this right here. But at 50 meters, obviously, that's not deep enough. I wonder if we change the cable to a check the because that's the original cable.
The subcons seal up better when there's pressure on them?
Yeah, I couldn't really give you a sciencey answer why, but a lot of times they do.
De lo que vamos en busca en esta expedición es específicamente de los microorganismos. La expedición está centrada en poder entender la ecología y la fisiología de los organismos más pequeños, que no los estamos viendo a simple vista, sino que vienen en esas muestras de sedimento o en las muestras de agua que vamos a tomar. Más allá de eso, también vamos a poder analizar las imágenes en busca de los macroorganismos, y también tenemos a bordo una observadora que va a estar registrando la fauna que se pueda ver desde arriba del barco.
What? Yeah. It gets finer further down, which is nice.
Yes.
Have you been able to see the last several cores? Yeah, that we actually pulled on the starboard side. So we lost one and repeated it.
Okay.
We have I think 3 really beautiful full length cores.
Okay. Do you know the number? You could know?
Yeah.
Okay.
Yeah.
El 10 fue el que se cayó y lo
El
volvieron a
10.
sacar, pero salió divino cuando salió.
Sí.
Era pura arena y se volcó, pero después sacaron otro muy lindo.
I have that 10, 11, and 12 are nearly full length. Does that square with you, Jimin?
Yes. Have you seen layers or differences on your? Okay. Okay.
By the way, if you think of sort of metadata that it would be useful, like for instance, full length core, half length core, etc., that we'd like to add to C-Log, let us know. The only advantage being that when you print out the automatic report, it'll be
Yeah,
there.
it'll be there.
Yeah. Yeah, you're the expert, so let us know what would be useful.
It's all the way out. Port you in so you can see with both cameras.
Go stretched out like
Go down low, sit. Yep. It's easy to use port too.
yeah, it makes sense stuck in my clothes.
Is that an all right spot, Jimin?
Oh, it's good.
That's just on the wall.
That made me bottoming out. Just on your control.
Just a wacky drop. Yeah, you see the zip line?
see. Maybe you understand the rookie core questions now.
Oh, no, it's
Didn't
all
work that way.
Happy here?
Yeah, let's next to any other core is good.
You want right next to it?
Could we? Is to the right of 7? Or what would you prefer? I just to figure you don't want those animals.
Yeah, next to 7 sounds good. Thank you.
Just so you guys know, the depth of these is dependent on when I hit bottom. It's not me randomly.
Yeah. We know. Here.
But he is pretty random.
All those things with push core.
Straight in.
Yeah, do you want me to, you guys need a photo of this or can I pull this straight out?
Jimin, do you want a photo of all the cores together, or can you just recover this one?
Look,
Go ahead and leave it there, please, and we'll go
yeah,
for a
photo-
frame grab.
photo would be great.
Thank you.
Yeah.
Los pilotos ahorita son Tyler, quien está controlando la arma o el brazo. No tengo una arma. Y Zach, quien controla el vehículo. Perdón, el manipulador, oficialmente.
Alright, you guys happy? Happy to pull?
Yep.
Yeah.
Yep.
Do we have a frame grab on that?
We do have a frame grab on
Wonderful.
that.
Thank you.
Okay, good pull. much for Sandy.
Whoever wants to break this. What wants to do? What's the option to do it? You have your
I do, but I don't know if I'm gonna have time today. I think we're over halfway done with this. All right, nothing crazy except obviously I got the armors draped down behind you right there, almost touching see that. And I've got 24% downs and auto heading on only. And as long as we don't move around, it should be fine. You can see the floats right there, right?
Yeah.
So we haven't moved. And you still got the heading where the current's just blowing you straight away. Okay? Okay. Donka.
Mark sample PC01, please.
PC01.
Looked at it.
Acá están preguntando si solo tomamos muestras de fondo y no, vamos a estar sí tomando muestras de fondo en todas las estaciones, pero además también del agua a diferentes profundidades.
Please mark PC02, a little less than half length.
PC02.
We've just changed pilots. Welcome to Claude and Mike. Claude on the vehicle, Mike on the manipulator. Okay, please mark sample PC04.
PC04.
Ahora estamos en la plataforma continental sobre Uruguay y recogemos muestras del sedimento del fondo del mar. Pregunta interesante sobre la formación del núcleo. Algunos biólogos que creen que la formación del núcleo es por una infección viral en un microbio, pero eso no es mi área de expertise.
Jacob,
Mark.
wants
Aye, thank you. What's that?
you would, oh, you would.
Yeah.
Okay.
All right, PC 17.
17. Yeah.
Yeah.
Switch.
Yeah, I can do that. Is there a question about it?
Yes.
Arc PC03, please.
PC 03.
Yeah, someone asked about the origin of the nucleus. That's one of the biggest questions that we're trying to address. There's at this point there isn't a whole lot of like clear evidence to support any one hypothesis of that, except the probably to me the most compelling is that the origin or the nucleus in eukaryotic cells evolved from a viral infection. But again, this is just entirely based on, as there are a lot of things like deep evolution. There's a lot of ideas, and oftentimes those ideas aren't really supported by any clear evidence. But it's so we're actually going to be looking at viruses as well, and those we're looking at viruses associated with Asgards. So we hope that we'll have some evidence to support one
Large
idea or
sample,
another.
PC05, please.
PC 05.
Good question, thank you.
Estamos en el mar de Uruguay, más o menos enfrente a la laguna del Sauce, en Maldonado. Pero bueno, muy adentro.
Thank you for the questions. That's a bunch of great cores here now.
It's like a lot of calcareous material in the lower part. Oop, oop.
Y sí, las muestras se están tomando con el Sebastian, que ya muchos conocen, es el mismo equipo. Lo que pasa es que nosotros solo vamos a sacar muestras de sedimento, entonces no vamos a ver en acción los bracitos colectando organismos, macroorganismos. Vamos a sacar solo agua y sedimento, pero es el mismo equipo.
Oh, yeah, it looks like.
Yeah, what is it?
Do you think it's an elasmo break? It's going awfully fast. Mark sample PC06, please.
PC06.
We're very excited about these samples. Our hunch is that these will be one of the best to look for these Hodarchaea. And the Hodarchaea were actually identified to be close to eukaryotes by Laura M., who's actually in the room with us. She's at the University of Rhode Island. You want to say hi, Laura?
Hi everyone.
How did you figure out that they're close to eukaryotes?
Oh yeah, so that was a very long process, but so everything starts with the kind of work that we're doing here, and so the idea is that after we do those samples, we extract the DNA from the organisms that are in those samples, and then we sequence that DNA, and we basically we look at that DNA and compare the DNA from all the species that are in those samples, plus all the species that we have sequenced in the past, all the species that we know on Earth if we summarize. And by comparing this DNA, the idea is basically that the closer the species are related, the more similar their DNA looks. So that's a very sort of simplified version of what we do, but that gives us an idea if we do that with all the species that we know on Earth and the ones we discover on a daily basis, we can basically reconstruct the genealogy of all the species on Earth. And this is work that we've done with Brett Baker, who is the chief scientist on the cruise here, and a lot of the people that are involved on this cruise, where we identified, we sequenced the genome from those new groups of Archaea that Brett mentioned earlier, including those Hodarchaeales, and by comparing their genomes to eukaryotes, we realized that they were among microorganisms, they were the ones that resembled the most the genome of eukaryotes. So they were our closest relatives that we know of on Earth so far.
I just want to verify. You can proceed, Mike. I just want to verify that this pore water core is long enough for you. Is it? It's about half length. That's as far as it would go.
Oh,
18. 1/8.
I'll get whatever
Okay, because we could potentially deploy it somewhere else, but you wouldn't have a visual on how close it is to anything. Right, because we've recovered everything else already.
Yeah, if there's time, we can maybe try pulling it out right next to it.
So are you saying that you'd prefer to dump the core and try to take a full length one?
If there's time, yeah.
Okay,
But
great.
if not,
Which one or the other one? Green one?
the other.
This one, number 08, please recover. Yeah.
Para quienes recién se suman y están consultando sobre dónde estamos y cuál es el objetivo de esta campaña, estamos en las aguas uruguayas y el objetivo de la campaña es poder encontrar a nuestros ancestros microbianos, a un grupo de organismos que fue recientemente descubierto, pero que se postula que son nuestros ancestros más cercanos, en el sentido que a partir de la asociación de estos organismos con otros se generó la célula eucariota, que es la base sobre la cual después se generaron todas las formas de vida que podemos imaginar, todas las plantas, los animales, los hongos. Y el objetivo de esta expedición es poder encontrar a los descendientes de esos primeros organismos y poder estudiar si en este momento, por ejemplo, están ocurriendo esas asociaciones que puedan llegar a estar dando origen a nuevas formas de vida. Sí, es interesante la pregunta acerca del nombre. Efectivamente, estas arqueas, que es el grupo grande al que pertenecen estos organismos, se descubrieron en el Ártico, en una zona que se llama el castillo de Loki. Loki es uno de los dioses de la mitología nórdica y es como el dios de la travesura, del engaño. Y a partir de ahí se descubrieron muchos otros linajes de estas arqueas, a los cuales se los denominó colectivamente como Asgard, que Asgard justamente es como el conjunto de dioses nórdicos. Entonces cada uno de esos linajes tiene un nombre que homenajea a alguno de estos dioses. Y bueno, tal vez aquí en nuestras aguas podemos llegar a encontrar un linaje completamente nuevo y tendremos que nombrarlo. Así que si les interesa participar de esto, pueden enviar propuestas acerca de cómo podríamos nombrar los nuevos linajes que descubramos en nuestras aguas.
PC08. After we get these cores on board, do you want to do any section or good? We have our hands full.
I don't think we can section three. We'll cover that.
Great. Sure.
What do you want? What do you want about this one? Do you want to try again?
We want to try again. How's it look, Jimin?
Yeah, just to the
Down
right.
it is.
Do you want to dump that core, Jimin?
Yes.
Please dump it, but maybe dump it away a little bit. And then we'll try to core as close to the original site as possible.
Can I ask you one question?
Yes, great.
So besides being a shallow dive, this is ultimately going to be a pretty short dive. So we are coming up on the end of the push cores, and we're probably not going to scoot around too much down here.
You want to guide him, Jimin?
Yes, that looks good.
But I
Whatever would be
do plan to do the ascent, as short as it is.
Okay. Right in there is fine?
Yes.
Okay.
Thank you.
All the way down?
Yes. Yes, please.
Jimin will be analyzing the chemistry of the water in between the sand grains in this core. He's a biologist from UC Santa Barbara in California. A bunch of them have that.
What do you think it is?
I think it's like calcium carbonate material from either like worm tubes or some other animal that's been been buried, but just a hunch. We'll find out.
It's almost certainly not marshmallow cream, right? Very low chance, but not. a
You
problem.
never know. I mean,
Could be though.
Ian and I live in the home of marshmallow cream.
Yeah.
Yeah. So,
Where's the home of marshmallow cream?
Somerville, Massachusetts.
That's just where they make it, or?
That's where it
they
originated.
Invented it, yeah. There's a festival every year called the Fluff Fest.
There was a sub I used to work on where there was a part number for marshmallow cream, because there were these bearings that you had to put painted on and stick them on it, and then when it get in the water, it would dissolve away, sort of like a glue. But there was a guy that would always eat it, so you go to do them and it would be gone.
We should get independent verification on this.
Yeah, it would be.
Jacob?
1-1.
Okay. How long does it take to go up?
From distance, probably
Yeah.
20 minutes,
20 minutes?
something like that, because we just
Okay.
gotta do the floats is all, right?
Okay.
We're pretty much there, it's just taking the floats on.
Okay, please mark PC18.
PC18.
Before we leave bottom, we let that dust settle just a bit, and then we're going to fire niskins 4, 5, and 6, please.
And that's then we're heading up, right?
Yep. I don't have anything else on my plan.
Keep up, there's more. That's the shortest dive in one time.
The shortest one that
Wait,
we've
hang
done?
on real quick. Jacob,
Really?
C-Log didn't log that PC18 when I hit submit. I don't know why.
I see it in here.
Oh, you do? Okay, it just didn't load on mine.
Okay.
Okay, sweet, sweet.
This is no light. Isn't that crazy? So this is just the
There we go.
sunlight. Still getting sunlight this heat.
Yeah.
So this is the
16.
Yeah.
ambient
Still.
light here
We
at
can
this
expect
depth.
sunlight at this latitude, probably between 150 and 200 meters.
Definitely over here.
Some amount. Do we have a waypoint on our parking spot here?
I could make one if you want.
I think that'd be
No, they don't let me.
nice for bookkeeping, please.
Sure. You got that time? Either one, as long as they can get the X or Y.
Yep. Which, of course, we've written down on the paper, but.
It does take a photo.
Sometimes redundancy is good, sometimes.
Department of Redundancy Department.
That's us. Yeah, it's orange. Six. This is 862.
Black and white.
Yeah. So this is a great example of how light works as you descend in the ocean. Because water scatters blue light and absorbs red light, you lose the warmer colors first. Which is why that sea star on the left of the screen, which when we had the lights on is actually orange, looks just sort of grayish green, the same color as everything else. Basically anything that has red in its pigments, that red will turn to black at this depth. What's the actual name on that? Is it marker 1?
I did landing spot 862. But we can do whatever.
That's great.
You guys. Actually, that would
All right, I think we're ready to fire niskins.
Okay.
Please.
Turn on the lights again. Definitely clears up that turbidity. Here we go.
Hello.
Hey, we got you. Hey, we got you.
How's it going? Let me know when you guys are ready. I think we're still waiting on seconds.
Sure. We just fired the last 3
All the
niskins.
cores are collected. We're doing the niskins.
Should be only a moment.
The other 3 niskins. They're gonna come up.
Okay. That is
Just
that.
one. Yep. It's not a lot of heterogeneity in the
Hop up.
mud around.
Brett, can we ascend?
Yeah.
We fired off the rest of the niskins. Let's come up, please.
Do it.
Go. Yeah. I can still come up off. Yeah.
You just sleep off.
Oh. Can you hold a moment, please?
You talking about pH?
pH.
Yeah, sorry. I was about to say
yeah.
something.
So where did we get the pH reading?
You know, Al,
Did-
otherwise we might have to get it empty.
Oh, did we turn it on?
No,
It's
been
on.
Yeah,
Okay,
it's been
good.
on the whole time.
Yeah, good,
But
good.
where they're piping it right now, besides recording it, I couldn't tell you. We'd have to get an empty for that.
Yeah,
Get 'em.
it doesn't show up in the C log. But
Can you see it anywhere over there on your monitors?
I pull up.
Hmm.
Yeah, we can get them though.
Just down the street to the left.
Oh, yeah. Might just
If
sit
it
down.
changes a whole lot, something's wrong with the ocean.
Might just sit down.
All right, that's the seabird pH.
Yeah.
Can we do an environmental observation, please?
Yep.
And put the pH in the comment.
Yeah, totally. No, everyone's. I have the raw counts if you want to.
That's
This
no,
is
that's
the correct
good for
count.
now. Thank you.
Wait, sorry, what is it?
8.07. Our ambient temperature is 11.1. Okay, our work is done.
All right, we're coming off bottom here, guys.
Yeah, Roger. You want to release control to me?
Sure.
Okay, taking control. Enabling joystick. One. Okay, coming up.
Copy that.
All right, that is us off bottom from dive 862. Pretty shallow, pretty quick, but we got our push course and we're headed back up. We do have a decent amount more dives planned for this cruise, but they are going to be push core heavy. On the deeper dives, we're expecting to kind of scoot around a little more, but on these shallow ones, we kind of got to just settle down and grab our cores. Gonna leave the cameras rolling until we're on surface, but they are already working on getting those floats because they never actually submerged all the way. So interesting dive in that regard.
No, not at all. Crazy, I tell you, crazy.
Probably step back a little, if you can.
You talking to me?
So thank you for joining us on this dive,
Roger that back.
and we'll see you next time.
Yeah.